Hello class, I'm Mr. Benjamin Corey Tusha, and today we're going to review my Lake Pleasant microcosm project for April of 2024. The objective of my microcosm project was to use components from the local environment to create a micro environment that I could grow in my house and see how that would grow over a 7 to 8 week period. For this activity, I used a standard size mason jar, about 32 ounce mason jar. I went and collected lake water and local soil along with a plant that was growing in the water from Lake Pleasant. Lake Pleasant is an area that is about 20 minutes away from my house, so my wife and I drove out there, searched along the beach to find any area or any plants growing in the water. We did identify one weed growing in the water, but it actually was just along the shoreline. This is the plants I ended up collecting. I collected a total of about 24 ounces of water, soil, and the fauna, and put it into a sealed environment for the duration of the experiment. As you can see for the microcosm project, this is what it looked like on March 4th when we went out to collect the flora and the fauna from Lake Pleasant. I did speak with one of the workers for the lake who had explained that this year there'd been a lot less rain due to the decrease in precipitation. The water levels of the lake were a little bit lower. This is a man-made structure, and so as you can see, the lower water levels meant that we were closer to the area of gravel and rock that was used in this man-made structure. Additionally, the worker explained that there was a problem with zebra mussels in the water, and that some of the vegetation along the shoreline had been substantially reduced due to this problem. This is a picture my wife took of me as I actually was collecting the plant, which ended up being an invasive species. It was a weed that was growing in the water. And I was able to collect some of the sediment. It really was mostly rock in this area and sand, not a lot of good topsoil in this area or sedimentary soil. So it took about 4 days for most of the material within the bottle to actually settle and for it to become a little bit clearer. The bottle itself, or the microcosm, was placed in indirect sunlight. It has sunlight about 3/4 of the hours of the day, and the temperature was inside the house, so it was varying in between around 72 and 68. It was in a window. I didn't really realize this until the end of the experiment, but that window actually has a shade on it, and that shade has some ultraviolet deflection capabilities. It might have actually made it to where a lot of the light that was reaching this wasn't really the UV type of light that we needed. The stems in the first day of this activity were green. There was blossoms on the actual plant. There was dark moss that had settled in the bottom of the jar, and it appeared healthy. There was no consumers that I could find. I tried to look for zebra mussels along the shore and didn't really find any. The water had settled, and after the first day, the water did appear murky, but obviously once it started to settle, things seemed to be doing pretty good. And the roots were at this point in the ground of the microcosm. This is how the microcosm appears. You can see it looks green and healthy. All the stems look alive and vibrant. On the next week, the light remains the same, temperature remains the same. The one thing that did seem to change here is the appearance of the forest green color of the actual stems seems to kind of die down a little bit, and it turned a little bit more of a yellowish tint. The leaves got a little yellow to green hue on them. The dark green moss had settled to the which had settled at the bottom of the jar, was still there. Again, it still appears healthy. The water was slightly cloudy, and the roots had come out of the soil. And so at this point, the soil, about 6 days later, the roots had popped out of the soil. The soil itself was mostly tan and brown. Here's a picture of that on at that period of time. Angle doesn't really show the same thing, but it does show you that there was a little bit of a difference here. On the 20th day, we're still in the same location. Temperature is still the same. The sunlight is 3/4 of the daylight hours. The main difference was the stems appeared to be green and yellow. The water is murky with kind of a green hue, almost like the chlorophyll is in the water. The leaves themselves had a green and yellowish tint, and dark green moss had floated to the base of where the stems were coming out of the water. There seemed to be a black film at the top of the water. It appears that the autotrophs, the plants, are actually starting to look very unhealthy at this point in time, and the water is becoming murkier each day. Here's a picture of what it looks like. As you can see, the picture here shows that the stems don't look very healthy, and you can see areas of separation where there's different colors showing up in the soil here. By the 26th, the conditions seem to be deteriorating. Black lines formed around the bottle or the bottom of the jar. It looks like the plant is turning into kind of like a blackish sludge. It is literally just kind of wilting in the middle of the jar. The moss has grown around it or is clumped around the middle of the plant. There looks to be like a black film on the top. There is black sediment in the very bottom, and the soil itself has got clear lines of what looks like gray material, and then there's a separated area of brown and tan as well. Overall, the water appears to be light gray or almost a blue-gray color. Here's what it looks like in direct sunlight. It's really not looking that healthy. On the 31st, a couple of days later, it really looks like this microenvironment has died. It looks like the plants are all dead at this point. The water has a hazy bluish gray color. There is a black tint near the top of the water and around the plant. It looks like that's where the moss has just kind of settled around the stems. The stems themselves have started to turn into yellow and bright yellow areas. There is still some dark green parts every now and then, but it looks more like it's turning into a black color. Again, it just doesn't look healthy at all. Here's what it looks like. You can see that it's very hazy in certain areas, and there seems to be kind of a sludge just right there at the top of the picture. On April 5th, the plant had been moved outside to see if we could get it into direct sunlight, see if this would change. The temperature range outside is going to be anywhere from 38 to 91 degrees Fahrenheit. It is Arizona. It is got some wide ranges of temperatures throughout the day. Two of the stems popped back up and actually started, the color kind of returned to kind of a darker green color. And it looked like there was some type of life trying to revitalize inside the jar. Oddly enough, the bottom of the jar, which had started to look mostly black and gray, there was a gray and black area that was still there, but a clear brown area of sediment started to rise to the top and in the middle of the jar, and a lot of the black area along the line just kind of subsided. And the autotroph appears to be alive, but very unhealthy at this point. The water appeared to be kind of a bluish color at this point and hazy. Here's what that looks like. On April 14th, the last day of the real observation, Again, it's been in direct sunlight for the last couple of days. Temperature range around the same, and the water around the top had returned to kind of a blackish around the top. And it looks like the soil has predominantly got a black hue. There is still a little brown or tan area in the soil near the top of the soil, and the autotroph again appears to be dead. The roots do appear to be a little hazy or fuzzy, like they were trying to grow, but it looks like maybe there's some mold that's starting to grow inside the bottle as well. So there is life, just not the life that I expected. Here's what it looks like. You can see here in the plant, it is very hazy, and the roots themselves just look like a glob of some dark green to black object in the middle of the bottle. The microcosm experiment was unsuccessful in its original hypothesis. The hypothesis that it would be a thriving community, it just did not really work out that way. Although there was signs of um, life as far as like moss or mold that started to grow inside the bottle. So overall, the microcosm did fail. I think some of the failure was due to the soil composition or complexity. I don't think it had enough sediment and minerals in it to support life. The second thing is the type of plant that was collected wasn't truly an aquatic plant. It just happened to be an invasive species that ended up growing along the shoreline. The placing the microcosm in indirect sunlight, I think ended up making it to where photosynthesis did not occur and ultimately led to the failure of the program. There were several different things I learned about this. The first was the ecological organization and the ecosystem is a delicate balance of the fauna and flora that it must be balanced to sustain life. The second thing is soil composition and complexity is essential to the producer's growth and development. And if the soil is not suited to support the growth and development of the autotroph, that basically life won't continue. And then third is the biogeochemical cycles needed. They need to be balanced. So there needs to be the correct type of biodiversity in the different trophic levels supporting one another. So there is photosynthesis being carried out and also cellular respiration taking place within this microenvironment to where it is continuously providing food for both organisms and keeps those cycles rotating. And I did actually learn quite a bit from this. The last picture that's on the bottom here is an example of some of the projects that 72 of my students in my environmental science classes started working on. And I used the microcosms idea from this course to get them to work on theirs. I do expect to continue to work on developing this and testing it in different ways. I'm going to work on developing a balanced ecosystem and use it in my environmental science classes. We're going to ensure that the soil complexity will support the life and the biogeochemical cycles that are needed in a closed system to matter and open system to energy. And I would like to use this as a project that we use throughout my class, semester-long class for environmental science. I think it would get the students to engage a little bit more. I do think I need to put some insects in there, such as like some snails and maybe even some other insects that might dig into the soil, along with some other types of autotrophs as well, to see how it would kind of develop over the course of a 90-day semester. I did learn a lot from this and felt like I'm taking a lot of information away from this program.